Okay, today we'll discuss the development of the second week of development in case of the fetal life. As we know, we'll be talking about this development starting from the 0 day, that is the day of fertilization. So we have talked for 7 days. Now we'll talk about the further development, that is development taking place between the 7th and the 14th day. So before we proceed further, we should know some terminologies like the germinal period. So germinal period is from the first to third week. Okay. So because this is the development in the very early stages when we cannot make out whether it is an embryo or a fetus. Okay. So this period is called as the germinal period from the first to third week of development. Okay. From the fourth week to eighth week, we call this developing structure as embryo. Embryo. By the fourth to eighth week, we call it as embryo. Then by the third month, that is the ninth week up to the ninth week to the ninth month, we call it as fetal period. So there are three different terminology which we use. The first to third week, we call it as the germinal period because we cannot exactly say what structure is developing. Okay. By the fourth to eighth week, we call it as the embryo, and the structure is called as embryo, and the period is called as embryonic period. And from the ninth week, that is the third month, up to the ninth month, we call the developing structure as the fetal fetus, and the period is called as fetal period. Okay, so these are the three important things we should remember. Okay, so the we have seen the implantation of the blastocele. This was the blastocele. We had seen that this is called as the outer cell mass, which becomes later the trophoblast. Then this is called as the embryo proper inner cell mass, which is becomes the embryoblast. Okay, so these cells outer, these are the trophoblasts which help in protection and providing nutrition, and the inner cell mass which become the embryoblast will develop into the embryo, and there is a cavity formed within that. This is called as the blastocystic cavity, or it is also called as blastocele. Okay, and the whole structure is called as the blastocyst. Okay, so now this blastocyst, which was covered by the zona pellucida, now the zona pellucida disappears. Now these tropoblastic cells have the very strong capacity to erode the endometrium. If you see here, this is the inner lining of the uterus of the mother. So now this trophoblast will go and get attached, and they start eroding this wall and getting inside. Now, if you see here in the next stage, the whole of the blastocyst has entered into the wall of the uterus, into the endometrium. So it has the capacity to erode, as I said, the wall and get inside the wall of the uterus, that is within the endometrium. So the implantation, as you know, implantation occurs on the 6th or the 7th day after fertilization. So this eroding of this blastocyst and getting into the wall of the uterus, this is called as the implantation. So this occurs on the 6th or the 7th day after fertilization. Then the site of implantation, as we already told you before, usually it will be on the posterior wall near the fundus of the uterus. So in case of the first, second, third pregnancies, it will be usually on the posterior wall near the fundus of the uterus. Fundus is on the top of the uterus. So, but with multiple pregnancies, it comes below and below. Okay, implantation becomes lower down. Now coming to the cells of the inner cell mass or the embryoblast. So these are the embryoblast or the inner cell mass. Now these cells differentiate into two types of cells. So this inner cell mass or the embryoblast, now it starts differentiating. Till now it is just a mass of cells. Now it slowly starts changing itself, and it is trying to become an embryo. So this embryoblastic cells, now they differentiate in two types of cells. One is on the top, that is called the epiblastic layer. On the top, these blue colored cells, which are, so initially they were all one. Now it is differentiating into top cells. These are called the epiblast, and this layer is called the epiblastic layer. And the cells are columnar cells, tall columnar cells. Below this yellow colored line, if you see here, the cells change themselves into hypoplastic layer, and this is called a hypoblast. And the cells will be cuboidal in shape. Cuboidal. You know the difference between the cuboidal as well as the columnar. So the upper cells, epiblastic cells, become tall columnar, and the hypoblastic layer becomes cuboidal. So now this embryoblast has changed itself into two layers, epiblastic layer and the hypoblastic layer. Epiblast, blue colored, and hypoblast, yellow colored cells. And by the 8th day, the tropoblastic cells covering the embryonic pole adhere to the endometrium and starts to proliferate rapidly, and they also differentiate into two layers. Now, as we have seen, these are the tropoblastic cells. Now they start eroding the wall. Now, once it is getting implanted, now they also change into two types of cells, just like the embryoblast change into itself into epiblast and hypoblast. Even the tropoblastic cell layers will change into two types of cells. One is called as the syncytiotropoblast, and the second is called as cytotropoblast. So this is cytotropoblast, and this is called as syncytiotropoblast. So they differentiate into two layers, and inner cytotropoblast, they are mononucleated. Mononucleated means with a single nucleus. Clear cells, clear cell wall, you can see easily the wall. So each cell can be seen with a nucleus and a wall around it. Okay. Then mitotic figures can be seen, which indicate they are dividing. They divide and migrate into syncytiotropoblast. So they keep dividing and they move into the syncytiotropoblast to make it a bigger layer. Now this is the syncytiotropoblast. What is this? This is an outer syncytiotrophoblastic layer, which is multinucleated zone without distinct cell boundaries. If you see here now, there are no cell boundaries like here. Here there is clear boundary and you can make out each cell with a nucleus. But here there is no boundary. So they look this because there is no boundary, so they look multinucleated. There are many nucleus without any cell boundary. So they are multinucleated zone without distinct cell boundary. So there is no cell boundary, so you cannot make out which is that cell, each cell. And there are no mitotic figures, no mitotic figures. So the syncytiotrophoblasts extend finger-like processes which secrete proteolytic enzymes that erode the endometrium, creating a hole through which the blastocyst is gradually buried into the endometrium. This syncytiotrophoblasts will start producing finger-like projection. If you see here, it is not smooth, but they are making finger-like projections which will erode the endometrium. They start eroding the endometrium by an enzyme that is the proteolytic enzyme, which will erode the endometrium here. So erode the endometrium and get inside the wall of the uterus. This is the wall of the uterus. This is called as the endometrial stroma or the endometrium. And here you can see uterine glands. Okay, so this is the lining epithelia which has been eroded. Now it is getting inside the wall of the uterus. By the 9th and 10th day, the hole in the endometrium is closed by a fibrin plug. The endometrium regenerates by the 12th day. So if you see here, so there is a big hole formed because of the eroding of this endometrium by this tropoblastic cells. Now slowly this hole becomes smaller and smaller. Later we'll see that, and there will be just a fibrin plug. Later we'll see, I will show you. So here there is still bigger layer. Now if you see here, there is a fibrin plug. So it has got inside the whole wall. Now this hole becomes smaller and it is closed by a fibrin coagulum or the fibrin plug. Okay, so this is what we can see. So there are two layers within the embryoblast, that is the epiblast and hypoblast, and there are two layers in the tropoblast. So this was the trophoblast. Now it has become two layers: syncytotrophoblast, this one, and the cytotrophoblast. Cyto means cell. When there is clear boundary, then it is called as cytotrophoblast. Syncio means joining together. So when they join together and they look multinucleate, then it is called as syncytotrophoblast. So both the layers, embryoblast and trophoblast, both have changed in themselves into two layers. Now what happens further? Now, now we talk about this two-layered structure, that is now it is called as the bilaminar germ disc. As I said, this period is called as germinal layer because still doesn't look like an embryo. So it is called as germ, and it is like a disc. So now if you see here, it is like a disc. So that's why it is called as germ disc. So it is two layer. So it is called as bilaminar because it is two layer. It is called as bilaminar germ disc. The area of contact between the embryonic ectoderm and endoderm is a disc-like, as we saw. This is disc-like and form the bilaminar germ disc. So it is two layers. So it is called a bilaminar germ disc, which will give the embryo proper. Later, it develop this two-layer structure. Disc will develop into an embryo proper. Okay. The embryonic disc will be oval in shape. If you see here, it is oval in shape. It is not round, but it is oval in shape. And lastly, pear shape, where the large end of the pear is at the cranial end and the small caudal end. Later, I will show you how it looks like a pear shape. Okay. When we see from above, it looks like a pear shape. When we see from the side, this is the side view, it looks like oval. But when you see from above, it looks like a pear shape. Now, the inner cell mass differentiate into two groups: the epiblast, that is the ectoderm, and the endoblast, or the hypoblast, which will differentiate itself into the endoderm. So epiblast will form itself into ectoderm. The whole body, our body, is made up of ectoderm, endoderm, and mesoderm. There are three layers from which all the structures are derived. Ectoderm means outside. Ecto means outside. So ectoderm means the outside layer. Okay, which will include the skin, the dermis. Okay, epidermis. Okay, the endoderm is the inner line. Okay, so now it is differentiating itself. The epiblast will now, it will be differentiated itself into the ectoderm, which is in the dorsal surface. The cells of the outer or the tropoblastic side of the embryonic blast form the epiblast. Now these are called as epiblastic layers, or the ectoderm. Between the epiblast cells, some vacuoles appear and coalesce to form the amniotic cavity. Now if you see here, just above that there is a small cavity formed, which will be initially multiple small cavities. Now they all join to form a small cavity called as amniotic cavity. Very important cavity form here, amniotic cavity. The floor of the amniotic cavity is formed by the columnar cells called as the embryonic ectoderm. So the floor, this epiblast, now it is called as the embryonic ectoderm. Flat cells line the roof of the cavity and called as the amniotic ectoderm, amnioblast. If you see here, it has a roof. This cavity, now amniotic cavity, has a floor which is made up of the ectoderm, and above cells it form a roof around that, that is called as the amnioblastic layer, amnioblast. So the cavity is called as the amniotic cavity, and the cells above that, that will be called as the amnioblastic cells. Now going further, so the hypoblast, that is the cells which are below. So here you can see here, this is the epiblastic layer, which is tall columnar. Above that there is a cavity called as the amniotic cavity, and above that we have the amnioblastic cells. Now we are talking about this layer, that is the hypoblastic layer. The hypoblast, which is the primitive endoderm or endoderm, a layer of cuboidal cells facing the blastocele. Here, this cavity, as we said before, it is a blastocele. This cavity is called as the blastocele, as we had seen before. So here it is the blastocele. Now, what happens to the blastocele? Now the blastoceles start changing. Okay. The blastoceles become lined with a layer of flattened mesothelial cells called as the excelomic membrane or Hauser's membrane. Now, this hypoblastic layer give rise to a thin membrane around this blastocele, this yellow colored structure. Now this layer is called as the excelomic membrane, or it is also called as the Hauser's membrane. Hauser is the scientist who found it. Okay. And this, now the blastocele is not called as blastocele because it is lined by a membrane called excelomic membrane. So this membrane, this cavity is called as the excelomic cavity. Excelomic cavity, or it is also called as the primary yolk sac. Very important structure. You have to keep watching this, how they keep changing. Okay. So initially there was only one cell layer here as well as here one layer. Now it become double layer here. Also here also it become double layer. Then there is a cavity formed here above, and now the cavity below is also lined by membrane, and there is a new cavity formed called as the excelomic cavity, and this is called also called as the primitive yolk sac. Later, the excelomic cells become modified and the cavity is called as the primitive yolk sac. Primitive yolk sac. Okay, so this is called as the primitive yolk sac. Later on, flat endodermal cells migrate round from the hypoblast to replace the degenerated mesothelial cells, forming a smaller cavity called as the secondary or definitive yolk sac. Now, this cavity changes from the primitive yolk sac to the definitive yolk sac or the secondary yolk sac. We'll see how it will change. Okay. Now, if you see here, the extra embryonic mesoderm. Here was the extra embryonic mesoderm here. So here there was the excelomic membrane, and below that will be formation of the something called as the extrameric mesoderm. Network of tissue appear between the trophoblast external to the amniotic cavity and the primitive yolk sac internally. So there will be formation of a network of trophoblastic cells. Trophoblast cells. The extra embryonic coelomic formation. The extra embryonic mesoderm will be formed here between the Hauser's membrane and the cytotrophoblast. Then it start differentiating. There will be formation of small cavities here also called as the extra embryonic coelom. Extra embryonic coelom. Okay. So this extra embryonic coelom, till now here there was no extra embryonic coelom. Okay. There was only the excelomic cavity or the primitive yolk sac. Now between this excelomic membrane and this cytotrophoblast, now there is small cavity formation. So there are small cavity formation, and there is a layer formed called as the extraembryonic mesoderm, and within the extraembryonic mesoderm there are small cavities again formed. These are called as the extraembryonic coelom. So the extraembryonic mesoderm will have two layers: somatopleuric, which is called as parietal layer, lining inside the trophoblast outside the amniotic cavity. So amniotic cavity. Now, so where is the amniotic cavity? This is the amniotic cavity. Around the amniotic cavity and lining all the way up to here, there is formation of a layer called as the extra embryonic somatic mesoderm, which is also called as parietal layer. Then there is an inner layer also formed, that is called as the splanchnic pleura, or the splenic pleuric layer, or the visceral layer lining outside the yolk sac. So this is the yolk sac, primitive yolk sac. Now there is formation of a layer here. This is called as the extraembryonic splanchnic mesoderm. Extraembryonic splanchnic mesoderm. So now the mesoderm is initially formed, then there are cavities formed, extraembryonic coelom, and there are two layers, extraembryonic splanchnic layer and the extraembryonic somatic layer. So this is the somatic layer and this splanchnic layer. Now this extraembryonic coelomic cavity is all joined together to form a larger cavity. A big cavity is formed. Now this is called as the extraembryonic cavity, or it is also called as the chorionic cavity. This whole cavity, which was small here with multiple cavities, they all join together to form a larger cavity. And this excelomic cavity, that is the primitive yolk sac, now it becomes smaller and smaller, and now it is called as the secondary yolk sac. Very important structure. And here still the embryo is in the form of two layer bilayer disc, and above that the amniotic cavity is still there. Okay, so now this whole cavity which was well connected, now it is all covered by this big cavity called as extraembryonic cavity or chorionic cavity, and it is only connected by a small structure called as the connecting stalk. Connecting stalk. We'll talk about that. Now the extraembryonic mesoderm, the cytotrophoblast proliferate to give a layer of loosely arranged cells called as the extraembryonic mesoderm, which fill the blastocele. If you see here, so suddenly there will be formation of the extraembryonic mesoderm, which join together. There will be formation of small cavities called as the extraembryonic mesoderm with the small cavities called as the extraembryonic coelom. Spaces appear among the cells of this mesoderm and become confluent to form a larger cavity called as the extraembryonic coelom or the chorionic cavity. So all these small cavities, which were small, they all join together, all these cavities, to form a larger cavity called as the extraembryonic cavity, or it is also called as the chorionic cavity, which divide the extraembryonic mesoderm. It divides the extraembryonic mesoderm. Now it is two layers still there. The splanchnic layer surrounding the yolk sac. So this is the splanchnic layer still there, okay, which is covering the yolk sac, and the somatic layer line the trophoblast and cover the amnion. So this is the somatic layer, which is called as the extraembryonic somatic mesoderm, okay, and this is called as the this thin layer here, this is called as the splanchnic layer, extraembryonic splanchnic layer, and this is called as the extraembryonic somatic layer, okay. So the splanchnic layer will cover the yolk sac, and the somatic layer covers this whole cavity, okay, line the whole cavity, even the amnion also. Here is amnion, so it will be covering even the amnion. Now the connecting stalk, a connection between the amniotic cavity and the cytotrophlast and will form the umbilical cord. So now there is only a small connection. The whole cavity, this whole cavity is floating in a bigger cavity called as the extraembryonic cavity or the chorionic cavity, and there is only a small connection between the uterine wall or the cytotrophoblast and this cavity, and this is called as the connecting stalk. Later, this connecting stalk will become the umbilical cord. Umbilical cord. So a lot of changes will be taking place during this first 3 weeks of development. Now, if you see here further, okay. Now, if you see, there is something called as decidua. Decidua. So, what do you mean by decidua? The uterine endometrium after implantation of the embryo is called as the decidua. We have seen the endometrium. So, as I said, the wall of the uterus is 3 layer. Outer layer is called as perimetrium. This muscular layer is called as myometrium, and the inner layer, this is called as endometrium. So this was the endometrium. With the implantation of the embryo, now this is called as decidua. Decidua. Okay, so this implantation, with the implantation of this embryo, now this endometrium is called as the decidua. So do you have any doubts till here? Students, do you have any doubts still here? Okay, if you don't have anything, we'll continue further. So this endometrium, as I said, after the implantation of the embryo is called as the decidua. So decidua is nothing but the endometrium after the implantation of the embryo. Okay, so here the embryo is developing. It is highly vascular and highly secretory to nourish the embryo. As we know, the endometrium, you might have already studied in the histology that it is highly vascular. And in the last class, we have seen there are coiled arteries within that, spiral arteries, as well as coiled the glands also. So they will be highly vascular. This endometrium is very highly vascular so that it can nourish the embryo, as well as highly secretory, so to give the nourishment to the developing embryo. Okay. After the implantation, the features of the endometrium are maintained and intensified. So with the implantation, the endometrium develops further. If there is no implantation, that will shed at the 28th day, as you know, as the menstrual cycle begins. So all the endometrium will shed. But with implantation, there is no shedding. Further, the endometrium will develop. as decidua. Okay. The cells enlarge, become vacuolated with glycogen and lipids, and these changes in the stromal cells are called as decidual reaction. So all the changes which are taking place within the endometrium with the implantation of the embryo, now it is called as the decidual reaction. Decidual reaction. Okay. Now the decidua itself differentiate into three parts. Differentiate into three parts. Then there are Deciduea basalis, Deciduea capsularis, and Deciduea paritalis. Deciduea basalis, where the part of the decidua at the embryonic pole, where the placenta is to be formed, that is called as the Deciduea basalis. Where the embryo get attached, where it is implanted, usually, as I said, it will be near the fundus in the posterior wall. So that part of the decidua will be called as the Decidua basalis because it forms the base which will in future develop into the placenta. See here, this is the placenta formed here. So this decidua will be called as Decidua basalis. Then deciduea capsula is part of the decidua at the embryonic pole facing the uterine cavity lumen, that is called as the deciduea capsula. A part of the decidua covers, because as we know the embryo has invaginated into the wall of the endometrium. So a part of the endometrium covers superficial, it forms a capsule. So this is called as the decidua capsularis. Decidua capsularis. And the remaining part, the remaining portion of the decidua will be called as the decidua paritalis. Decidua paritalis, where it is not implanted, it's called as paritalis. So decidua basalis, where the embryo is implanted, part of the decidua covers the implanted embryo, so that is called as the decidua capsularis, and the remaining part, this is called as the decidua paritalis. Later, with the development of the fetus, if you see here, this decidua capsularis and the paritalis, they both get fused. So as the embryo starts developing, becoming bigger and bigger, both these layers, deciduea capsularis and the paritalis, will fuse. Okay, so here you can see there is no differentiation between the paritalis and capsularis. Now there is something called as chorion, a very important structure developing, that is called as chorion. So what is this chorion? Chorion is the trophoblast with the underlying extra embryonic mesoderm. Trophoblast, what is trophoblast? We had seen trophoblast differentiate into two types, cytotrophoblast and the syncytotrophoblast. Okay. Now this trophoblast with the extra embryonic mesoderm, extraembryonic mesoderm together. This is called as the chorion. Chorion is formed of the syncytotrophoblast, cytotrophoblast, and the outer layer of the extraembryonic mesoderm. So you can see here, this is the syncytotrophoblast, green structure, colored structure. Then this light blue colored structure, this is the cytotrophoblast, as you can see the cells clearly. And this is the extraembryonic mesoderm we had seen, the parietal layer, paritalis, or the parietal layer of the extraembryonic mesoderm. These three layers together, these are called as the chorion, or it is also called as the chorionic plate. Very important structure, because it will future develop. How it develops, we will see. Okay. It divides the mesoderm into an outer and inner layer, except a region called this connecting stalk, the body stalk. As we have seen, this is the connecting stalk. The latter will form the future umbilical cord. So this connecting stalk will later form the umbilical cord, as you can see here. So here there was no umbilical cord. Now it is developed into an umbilical cord from this connecting stalk. Now something is going on within the wall. Now we'll see in the midterm. We have seen the changes taking place here. Now we'll see what are the changes taking place here. Okay. The chorionic villi. There is something called as formation of chorionic villi. We have already seen the chorion. Chorion is nothing but the three layers here. The syncytotrophoblast, cytotrophoblast, and the extraembryonic mesoderm. These three layers are called as chorion or chorionic plate. Now there is something called as chorionic villi. The syncytotrophoblast is divided into finger-like chorionic villi, which are primitive invade the endometrium. If you see here now, Now this syncytiotrophoblasts, now it starts invaginating into the wall. As you can see here, this is the endometrium, and it has a highly vascular, so there are a lot of blood vessels. So now this syncytiotrophoblasts will give rise to finger-like projections here, finger-like projections into the endometrium, and it will form primitive or the invade the decidua. They start invading the decidua. Now there will be slowly formation of vacuoles. If you see here, there is formation of small spaces called as the vacuoles appear in the syncytiotrophoblasts. So they start appearing in the syncytiotrophoblasts with each other to give lacunae. So now these are called as the vacuoles. Now they are called as lacunae spaces that become confluent and form the intervillous space. So they all join together to later form the intervillous space. We will talk about that. So these vacuoles are formed here, or the lacunae spaces in between these finger-like projections called as the syncytiotrophoblasts. Now there is formation of something called as the primitive villi. Now these will form something called as primitive villi. The villi erode the decidual blood vessels, forming lacunae in the intervillous space. Now they start eroding the wall still inside, and as you can see here, it is highly vascular. Now they start eroding the blood vessels also. Now when they erode the blood vessels start leaking into these vacuoles. Now if you see here, these spaces are filled with blood. So now blood start entering these spaces called as the vacuoles. So the villi erode the decidua. So decidua is this one. So they start eroding the decidua, decidual blood vessels, forming lacunae in the intervillous space. So these are lacunae intervillous space filled with maternal blood. Now the maternal blood has entered into this villus. Fetus is still here. Fetal, which is in the form of bilaminar disc. Still it is in the bilaminar disc. Now the blood start entering into this intervillous space filled with maternal blood that helps to nourish the embryo by the diffusion. So still the blood has come until here, but still it cannot nourish the embryo because still there is a lot of space is there through which it has to enter. So now still it will nourish the embryo by diffusion, not by direct blood flow, but by diffusion. So till here the blood has entered. Now still it is nourishing the embryo. So this was all about the changes which are taking place in the second week of development. So we'll once again revise, we'll summarize what are the changes which are taking place in the second week. Okay, so the second week starts from the 7th day when it gets implanted, and by the 8th day of development there is differentiation of the outer cell mass, that is the trophoblastic cells, into cytotrophoblasts and the syncytiotrophoblasts. So now we are trying to revise once again before we end. So what are the changes taking place at each day? So by the 8th day, the cytotrophoblast has differentiated into two cells, cytotrophoblastic cells and the syncytiotrophoblasts. Trophoblasts are differentiated into cytotrophoblasts and the syncytiotrophoblasts. Okay, you can see here this is the cytotrophoblast cells and this is the syncytiotrophoblasts. Differentiation of the inner cell mass into epiblast and hypoblast, and the inner cell mass, which is the embryo proper, has differentiated into the epiblastic layer and the hypoblastic layer. And there is formation of the amniotic cavity. Also there is formation of the amniotic cavity just above the epiblast. So these are the changes taking place on the 8th day. On the 9th day, we have seen that there is the lacunar stage phase of trophoblast developing, where the lacunae are formed in the syncytiotrophoblasts. So the syncytiotrophoblasts started eroding the wall, and there is formation of the lacunae here, spaces. Then there was also formation of the primitive yolk sac from the hypoblast. As we had seen, the hypoblastic layer will give rise to a membrane, thin membrane called as the excelomic membrane or Hauss's membrane, which will chain the blastocele or the blastocystic cavity into excelomic cavity, or it is also called as the primitive yolk sac. So now there are two cavities, amniotic cavity and the primitive yolk sac. Now by the 11th to 12th day, there were many changes which were taking place. Okay, so the blastocyst is completely embedded in the endometrium wall. As you can see here, this is the the endometrium. It has completely entered the endometrium wall, and now there is formation of thin plug Okay, the whole of the eroded wall has regrown and there is a thin plug. Maternal blood start entering the lacunar system, establishing the uteroplacental circulation. Now, as we have seen from the lacunae, so this was the lacunae. Now it started eroding the blood vessels, and the blood vessels started leaking into these spaces. Okay, now this is called as the uteroplacental circulation, through which by diffusion, still by diffusion, the blood, this blood, maternal blood will be nourishing the fetus. Still there is no direct contact, but by diffusion it will be supplying the nourishing the developing fetus, which is still in the two-layer germ disc. So this is called as the uteroplacental circulation, this type of circulation. Okay. Formation of the extra embryonic mesoderm. Then there was formation of an extra embryonic mesoderm between this membrane here, which is called as the Hauser's membrane or the extra embryonic membrane, and the cytoplasm. There was formation in between. There was formation of a membrane called as the extra embryonic mesoderm. Now this extraembryonic mesoderm, as soon as it is formed, it started differentiating into extraembryonic mesoderm into splanchnic layer and somatic layer. Somatic layer was here. This is called as the extraembryonic somatic fluid membrane, and the inner layer is called as the extraembryonic splanchnic mesoderm. This is the splanchnic layer. This is the somatic layer. And then there was formation of small cavities here, formation of the extra embryonic coelom. So here there was formation of the extra embryonic coelom, which later changed itself into the chorionic cavity. Also there was changes within the wall of the uterus, that is called as the decidual reaction. By the 13th day of development, the surface defect in the endometrium has usually healed, as we have seen. So now it has totally healed. So the hole which was formed initially in the endometrium, now it has completely grown. If you see here, there was thin fibrin plug. Now it has completely grown. It is totally closed, and now it is within the wall of the uterus. Okay. Formation of the primary villi. With this formation, there will be villi-like structures. Okay, in between there is blood and there will be green structures. This is called as the primary villi. We'll talk about that later again. Formation of the secondary or defective yolk sac, definitive, sorry. So formation of secondary or the definitive yolk sac. This primary yolk sac, which was primary yolk sac here, now with the joining of all these extraembryonic coeloms, now these coeloms join together to form a very big cavity. Still the splanchnic layer and the somatic layer are still there, but this cavity has become very big, and this yolk sac, primary yolk sac, has become smaller and smaller. Now it is called as the secondary yolk sac. This is called as secondary yolk sac. This is the embryo, which is bilaminar disc. Now here is the amniotic cavity. Now this whole cavity started flowing in this cavity called as the extra embryonic cavity, or it is also called as the chorionic cavity. So this was all about the development in the second week. Okay. So if you have any doubts regarding this part, you can ask me, or the previous lecture or this lecture, you can ask me now.
Thank you, doctor.
Okay, there is a lot of information here. As I said again, once again, I repeat that all the lectures are related here. So you should understand, if you want to understand the second lecture, you should understand the first lecture. And if you want to understand the third lecture, which is today again at 2 o'clock, you have to read once again, go back. It cannot be understood in one go. So you have to again repeat, read it, and try to understand. And if you have any doubts, you can ask me later also. But read it before we go to the third lecture, which is again at 2 o'clock. So there is a very short gap, but still it is all related. If you understand this lecture, only then you can understand the third lecture. So because they are all connected. So it is first week of development, second week of development, then third week of development, which we'll cover this week. Okay. So if you don't have any doubts, okay, we can stop here. Thank you very much. Thank